I am now planning to explain to you in the simplest possible way the method of using infrared illumination in order to analyze the presence of molecules in a material. It is a method called vibrational infrared vibrational spectroscopy, commonly used in the chemistry labs. But we researchers in the electrotechnical area are also interested in this for various types of applied sensors. Here we can see the sun as the dominant source of illumination. And light rays from the sun is in this case passing through a thin material. And different material properties will give the light also different colors ranging from red to green. Colors that can be perceived by human vision. Alternatively, the light could be reflected into a surface, and the properties of the material where it is reflected is giving the light rays different colors also perceived by human vision. Now, the human vision has photoreceptors in the retina inside the eye. The first type of photoreceptor is called rods, and the rods are sensitive for low light intensities, but there is no possibility to sense different colors. But on the other hand, for more higher intensities, we have photoreceptors called cones. And cones are divided into three types of photoreceptors: sensitive for red color, for green color, and finally for blue color. And it is the intensity balance of the received light on the red, green, and blue photoreceptors that is enabling the human vision and the humans to perceive different colors, ranging also, including also purple and yellow or pink, those colors that are outside of the fundamental colors. Light can be viewed in two different forms. Either we can see it as an electromagnetic waveform that is propagating in this direction, having a periodic intensity in this way. And the distance between two maxima here, we measure it or characterize it as the wavelength and call it lambda. It can be the distance between two maxima or the distance between two minima. But equally, as an electromagnetic waveform, we can view it as a stream of particles. We call the particles photons. And each one of the particles has a defined energy level, E. And the energy level, there is a certain mathematical relation between the energy level of each one of the photons and the corresponding wavelength. And the wavelength of light that humans can perceive is ranging from 380 to 740 nanometer, ranging from the most intensely blue light to red light. The wave number is just another way of expressing the wavelength. Wave number is more the rate of the periods per unit of distance in the propagation direction. And we express the wave number according to this very simple formula. A diagram is shown here, and on the horizontal axis we see the wavelength of light ranging from 1000 to 1700 nanometers. This is infrared light, much longer wavelength than the kind of light that can be perceived by human vision. On the other hand, on the vertical axis, we see the intensity of the reflected light that has been reflected into a surface that we want to analyze using a method called spectroscopy. And the diagram is now showing the intensity of the reflected light distributed on different wavelengths. And the way we can have this distribution is that we are using an instrumentation that can divide the measurement on a series of spectral channels. And one spectral channel is very narrow, just representing a few nanometers, which means that the range of light here will be represented by hundreds of these spectral channels. The diagram by itself that is showing this kind of spectral profile for a material is what we call spectroscopy, the method of spectroscopy. And the spectral channel here, the very narrow spectral channel, it can be somehow compared with the photoreceptors that we have inside the retina of our eyes. It's only that for the retina in our eyes, we only use 3 types of receptors: green, red, and blue light. Here we have hundreds of spectral channels for the spectroscopy method. How do you think the world would look like if humans could see several hundreds of colors, also including the infrared light? I'm just thinking now. What makes the infrared region of light so interesting is that when it is radiated onto a surface, the molecules of this material that is irradiated, they start to vibrate. And you think of a molecule, it is made out of a number of atoms. Think of these 2 balls as 2 atoms. And they are connected with a spring, the binding energy of the atoms inside the molecule. And once you get the stimulation from the outside, from this light that is irradiated onto the molecule, it will start to vibrate. And the vibration energy tells us something about what kind of molecule we are talking about here. In this illustration, we see a water molecule. And you may remember that the water molecule is made out of 1 oxygen atom and 2 hydrogen atoms. And the hydrogen atoms, they are connected with a certain binding energy to the oxygen atom. And the vibrations that are caused by irradiated infrared light, it can be simulated using an online simulation tool. And on the left side, we see a graph, a diagram with a horizontal axis giving an indication of the wave number being almost the same as the wavelength. And on the vertical axis, we see how light is actually transmitted through the material, through the water. And here we can see 3 distinct wavelengths where the light is almost completely absorbed by the water molecule. And this absorption happens when the atoms of the molecule, they start to vibrate, and then you are losing all the energy into this vibration and into the material. The oscillations of the atoms within a molecule is depending on the binding energies between the atoms. And you may think of the binding energies that corresponds to the strength that I'm attaching to a guitar string. So if I'm increasing the strength, then the frequency of this vibration will also increase. If I reduce the binding energy or the power or strength of the string, then I will also reduce again the frequency. So then again, the different spectrum of vibrations of different molecules will create unique spectral signatures of each molecule that are present in the material. This is what makes the vibrational spectroscopy so interesting. Here we see again the water molecule from the previous example. And we can see that the water molecule is actually vibrating in 3 different modes, this way creating responses in 2.738, 2.745, and 6.499 micrometer, respectively. And this kind of analysis method from the chemistry lab, called vibrational spectroscopy, is very interesting for various contactless sensing applications. One such contactless method for measurements is the use of hyperspectral cameras. And you can think of a hyperspectral camera as a color camera that has hundreds of spectral channels for each one of the pixels. And the camera can also be made sensitive in the infrared region, such that those hundreds of spectral channels can be used to implement vibrational spectroscopy at every pixel. You see here a couple of images from our vision lab at the university. Here is a frame, and on this frame is assembled 2 cameras, hyperspectral cameras, as well as illumination. These are halogen lamps that also emit a lot of light in the infrared region. And on this moving table that is controlled by a linear stage, there are several of these plastic items that can be imaged by the 2 cameras. And from that data, it is possible to identify these image components and classify them according to what kind of polymer type this plastic is made out of. So it's a material classification in this case. So this kind of measurement setups opens up for a lot of different kinds of applications, and some of those that we have been looking into in our research so far is the, as you can see here in these pictures, the polymer classification, but also detection of plastic materials in food waste. And we have been looking into road condition monitoring, being able to identify a road pavement covered with ice, with water, with snow, or dry asphalt.